So the only sort of the odd crossing over is, again, between the Y chromosome and the X, because there's such a mismatch in size. And this crossing over only takes place within the pseudo-autosomal region, where there are genes that are homologous. And at least one crossover is required for correct separation later, in the later stages of mitosis, or meiosis. Okay, so what can go wrong? Well, without going into any details, this just shows you some of the things that can happen. You can lose part of a chromosome. You can either at the end or in the middle someplace. What else? I can't even see what these are. Or you can have unequal crossing over taking place. This unequal crossing over, by the way, often takes place at these repetitive sequences that I mentioned before. Some repetitive sequences are spaced, throughout the genome, and if there's a mismatching of a repetitive sequence between the two chromosomes and recombination takes place there, you end up with unequal recombination. Rings can form like this. It turns out, I'll mention this again later on, that the telomeres have a very special structure, molecular structure, which I'll show you later, and those protect the ends of the chromosomes. And if a chromosome breaks for some reason, there are enzymes in the cell nucleus that cause two broken ends to be fused together. And so if, as occasionally happens, the two ends of a chromosome lose their telomeres, they can form a circle and form a ring chromosome. And then you can just look for yourself. There are all sorts of other odd things that can take place. Now, sometimes rearrangements take place that are in themselves not harmful to an individual. So, for example, sometimes an inversion may take place where an internal segment of the chromosome undergoes an internal recombination, and the effect is not to eliminate the sequence, but just to invert the sequence. This happens occasionally. Now, if the points of inversion don't break any genes, or don't interfere with the expression of the genes, then for that person, the person who inherits this inverted chromosome, there's really no problem. The person is perfectly normal. The problem arises, however, when that person makes gametes. Because during meiosis, remember during meiosis 1, the homologous pairs of chromosomes have to align exactly. And if you have one that's normal and one that has a big segment that's inverted, well, the alignment becomes very complicated. And you get structures like this. And again, I won't go into detail here, but if you go over this at home, and you'll see that you can end up with chromosomes that are not quite the right structure. And so then individuals will, if zygotes form with these chromosomes, they're not viable. But occasionally, children will be born from parents who are carrying these inversions. And for example, they might be born with 2 copies of the A chromosomal region here, and then be missing the D chromosomal region, or just the opposite. And there are genetic syndromes that are known where the children are born, but they're born with genetic defects, and it's caused by this. Okay, so again, take a look at this more carefully at home. Here's another complicated slide, and the point here again is that this is an example where in the previous generation an unequal crossing over has taken place. But actually, even though these derivatized chromosomes have been created, there's no loss or gain of genes. And so for the person carrying these odd chromosomes, there's no problem. Often. There are no genetic defects. The problem occurs, though, when that person is making gametes, either sperm or eggs, because during meiosis 1, you get these very odd sort of alignments of sequence. And then unusual crossing overs that can lead to chromosomes that are defective. So this rearrangement took place in the grandfather or grandmother. The son or daughter of that couple has no problems, but their children may suffer genetic disorders. Okay, so again, I won't go over this in detail, but take a look at home and see if you can figure out what's going on. Oh, this slide, though, I want to introduce a notation here. This is sort of important. This is a genetic shorthand to describe what's going on. And the way you translate this is the T means translocation, and the translocation involves chromosome number 3 and chromosome number 11. And then the second part here tells you where the breakpoints are. So what this means is that there's a break on chromosome 3 at Q12 and a break at chromosome 11 at P15.5. Okay, so that's what this notation means. At the end of the slides, I have 2 pages that I copied from a textbook that describes more notation that's often used for describing chromosomal rearrangements. And it's just, you don't have to memorize this, but just take a look at that table. Because when you're reading the literature, or even as a doctor, you may be told that someone has this kind of disorder, and it's a good idea to understand how to read this kind of notation. Okay, so this is just an example of chromosome painting, and it shows the power of chromosome painting to reveal recombinant, genetic oddities. So here they've painted chromosome 1, 4, 2, and 4 yellow, and the rest have been stained red. And what this allows you to see very clearly is that this is chromosome 4 that has undergone a recombination with one of these other red chromosomes. And so part of chromosome 4 is stuck over here, and the other one is stuck. Well, you can see the recombination is taken. Part of the red chromosome is now attached to this chromosome, and the other one is crossed here. So you can see very clearly that some sort of recombination is taking place. And this shows the power of this chromosome painting technique. Okay, so then those are things that can happen during the process of crossing over. But there are also mistakes that take place that have to do with the segregation of chromosomes. So during meiosis 1, the homologous pairs of chromosomes, chromosome 1 pairs with the other chromosome 1, both of which have replicated. So each, you have 4 chromatids here in this complex. And in theory, the 2 homologous chromosomes should go into different cells. But occasionally, or not infrequently actually, that doesn't happen, and the cell undergoes division, but both of these chromosomes fail to separate, and you have which is called non-disjunction, and you end up with daughter cells that one contains an extra pair of these chromosomes and the other one's missing it altogether. And then when gametes are formed from these cells, you have 2 gametes with an extra chromosome, and then 2 gametes which is lacking a chromosome. So when these fertilize, if these are sperm and they fertilize eggs, then the zygote that forms from that is going to be have either an extra chromosome or be missing the chromosome. And so this same thing can happen at meiosis 2. So here you have a correct disjunction of these pair of chromosomes. They go in, one set goes into one daughter cell, the other goes into the other daughter cell. But then at the next step, this can also not separate correctly. And so you can have non-disjunction either in meiosis 1 or meiosis 2. And the effects are similar. Here, although the proportions are different. Here you have 2 normal gametes, and here you have 1 gamete that has an extra chromosome, and here one that is missing one. So if these are sperms, they fertilize eggs, then there would be a problem. I said sperm, but actually this is much more common during oogenesis, during production of eggs. It's about 10 times more frequently occurring there than in the production of sperm. So here's an example. I think probably everyone's heard of Down syndrome, right? Well, Down syndrome is known to be caused by trisomy of chromosome 21. And what that means is there's an extra copy of chromosome 21. And so if you look at the karyotype of an individual with Down syndrome, you can see that there's 3 chromosomes here instead of the normal 2. And it turns out that this can arise either from disjunction in meiosis 1 or meiosis 2, but disjunction at meiosis 1 is about 3 times more common. And again, it's about 10 times more common for this to occur during oogenesis compared to spermatogenesis. Okay, so here, this is a table, and again, I'm not going to go over this in great detail, but I'd like you to look at this carefully at home. I just want to point out, this is how frequent chromosomal abnormalities are. If this is a study that looked at roughly 10,000 pregnancies, and among those, 9,200 had normal sets of chromosomes, normal karyotypes, but 800 did not. There was some mistake. So this is 8% of the pregnancies have some sort of chromosomal abnormality, which is pretty high. That's pretty high. You know, it's like almost 1 in 10. The numbers below just show you how these 800 are distributed across different kinds of problems. You notice, though, that most of these do not result in live births. So a major cause of spontaneous abortion is the fact that fetuses that carry one extra chromosome or lack one chromosome tend to die in the womb or be aborted spontaneously. And it's only a minority that actually are born. So a minority of individuals, of babies, actually carry these. So there's some sort of, it's a very error-prone business of this separating the chromosomes. But there's some sort of biological mechanism at work that prevents too many of these to be born. Except for 21 trisomy, most of the other trisomies or monosomies are lethal. Occasionally, babies are born with trisomy 16, 18, or 13, but they usually die in infancy. So having an extra chromosome, except for 21, is usually lethal. Okay. Another thing that's sort of interesting, that's important to be aware of, is that the frequency with which these abnormalities occur increases with the age of the mother. And the increase is really dramatic. So that mothers who have their children early in life, in their teens or 20s, have a very low risk of having these non-disjunction events take place. But when you get up into the 40s, the rates are very, very high. This means 35% of the pregnancies of conceptions have this kind of defect. That's 1 in 3. So older mothers really need to be aware of this and of how dramatic this increase is. Now, most of these, this 30%, would result in spontaneous abortions, and so that the baby just would not be born, and maybe the mother may not even know anything about it if it occurs very, very early. For 21, however, these babies have a good chance of being born. And so this graph shows the rate of increase of babies with, fetuses or babies with 21 trisomy. And it's both measured as measured by amniocentesis and then at birth. And here, this would be 12%. So, whereas it's very low here. And so the rate goes up enormously as the mother's age increases. So nowadays, it's almost, most people consider it sort of almost mandatory to have, if you're thinking of having a child, when you're in your 40s or later, it's probably a very, very good idea to have a test done to see if there's trisomy 21, because there's a big increase. Now, I don't want to make you feel that women are responsible for all genetic defects. It turns out that men also have a problem, but it's a different type of problem, and that is that men are producing sperm throughout their whole life, and because of that, the number of cell divisions and DNA replications is very large, and because of that, the men tend to accumulate other types of mutations. And it turns out that most of the point mutations and small deletions and other types of mutations that come into the new baby come from the men, and that increases with age. It's also known that the offspring of older men have a much higher chance of developing autism or schizophrenia than younger men. So it seems like having a baby early might be good advice in general, but nowadays, of course, a lot of both men and women are under a lot of professional obligations, and so people postpone having babies. A friend of mine actually had his first daughter when he was 69, which is a little risky. And another friend of mine has a family of 5 boys. The last boy was born when the mother was 49. But healthy, absolutely healthy. So you certainly, even at these advanced ages, the majority of the offspring are healthy. You should remember that. But the risk is also significant. So when you're planning your families and you're in life, you should sort of take this into consideration. Okay, so now, so far we've been talking about chromosomal abnormalities that occur during the generation of germ cells, the sperms or the eggs. But chromosomal abnormalities also can occur in somatic cells, but usually at a much lower frequency. In cancer cells, however, this seems to be the rule. And so one of the hallmarks of malignant cancers is an abnormal karyotype. So here's a karyotype taken from a breast cancer cell line. And if you look at this carefully, you'll see there's grossly, the wrong numbers of chromosomes. There are 1, 2, 3, 4, 5 copies of chromosome 1, 3, and 2, and there's some very odd translocation event that's taken place here and there, and there, and there. So this is actually one of the bases by which doctors can decide whether a cancerous tissue is malignant or not, whether the karyotype is normal or not. And again, grossly abnormal karyotypes like this are characteristic of malignant cells. Okay, so then I'm just going to show you, I think you've just had a lecture that went over this, so I'm going to go over this very quickly. And we're just going to talk just for a minute or two about the molecular structure. So here's the chromosome here. It's actually easier to start with the DNA. So I'm going to start down here, and we're going to work up our way up here. So as I mentioned before, you have 2 meters of DNA in every cell. The diameter of the DNA is very small, however. It's only about 2 nanometers. So it's very thin, wire that extends for 2 meters. This is organized into nucleosomes. You just had this, right? The nucleosome contains, it's an octamer of 4 different histone proteins, or 4 histone proteins. And there's usually 2A, 2B, 3, and 4. And there are 2 each of those, so 8 altogether. And then there's a histone 1 that sort of binds the outside of this and stabilizes that. And so they're organized, and these are called beads on a string. So these are the nucleosomes. And then the nucleosomes are further wound up to make a fiber like this. And then those are eventually organized on some sort of scaffold. And then those scaffolds get compacted like this, and then eventually get a chromosome. Okay? So it's a very orderly way that, by which these very long molecules are packaged up. They would have to be, otherwise there'd just be an impossible tangle inside the nucleus. I think you've also heard about this, that the histones, chemical modifications of the histones play a very important role in changing the architecture of the chromatin. And this when we talk about the architecture of the chromatin, we're talking mostly about how open or compact or open the chromatin is. And again, heterochromatin is a state where it's highly compacted, and gene expression usually does not take place in those areas. Whereas euchromatin is a state where the chromatin is more open, and gene expression can take place. And there are many factors that determine whether the chromatin is heterochromatin or euchromatin, but one of the important ones is the acetylation of the tails of the histones, primarily histones 3 and 4. So just roughly speaking, when there's the histones tails, these are the amino terminal tails, are acetylated, it tends to be, the chromatin tends to be open, and when they're deacetylated, it's closed down. And there are enzymes that do that, so that there's a histone acetyltransferase called HAT, and so when it works, it tends to open up the chromosome and allow gene expression to take place. And then there are deacetylases that then can cleave these acetyl groups off, and that tends to send the chromatin into a more compact state. Now, in addition to acetylation, there's also methylation going on. And roughly speaking, the open chromatin, the euchromatin, tends to be less methylated on histones compared to the heterochromatin. It's actually quite complicated what's going on, but just roughly, acetylation is associated with euchromatin, and methylation and deacetylation is usually associated with heterochromatin. Okay. Now, another thing that goes on is at the DNA level, and that is that most of the human DNA is methylated on C residues, but only C residues that come in the sequence CPG. Okay, not all C residues. Specifically, that pair, and the methylation is on both strands, and in general, most of the chromosome is methylated. Not uniformly, but the majority of CPGs are methylated, except for it turns out that about half the genes in the genome have very GC-rich 5' ends, which means that the promoter region and the 5' end of the gene is very GC-rich. And because it's GC-rich, there are a lot of these GPC regions there. In promoters that are active, where gene expression is taking place, those CPGs tend not to be methylated. These CPG rich regions are called CPG islands, and it's generally true that for genes that are being expressed, there's less methylation there than in genes that are not being expressed. That's not the whole. For a long time, people thought that was the whole story, but it turns out it's not the whole story. It turns out that different tissues have different patterns of DNA methylation, and that occurs not only within these GPC islands, but also in regions outside of the islands. So it's sort of complex. Furthermore, we don't have time to go into this now, the pattern of methylation can be modified by the environment. So environmental influences can have an effect on the way the DNA is methylated, and that can affect gene expression. And very interestingly, it's been shown in mice that the environmental influences on the pups, the mouse pups when they're little, can actually affect their psychological state, how nervous they are, for example, for their entire life. So, and there's evidence from humans, actually, that these patterns of methylation that may change in response to, for example, famine, starvation, can actually be transmitted across generations. So that, for example, there's studies where a population in Finland that suffered famine during the grandparents' generation, that the effect of that is to increase heart disease and other diseases in the children and grandchildren. So this is something that's just recently been discovered, and it almost sounds. You know about Lamarckian. Have you heard about if I say Lamarck and Darwin, does that make Darwin showed that that evolution takes place through a selection process, where the survival of the fittest, the changes that have occurred during evolution are acquired by that method. Whereas an older idea is that environmental influences change the genetics. So the old Lamarckian idea was, for example, giraffes may develop long necks because they have to stretch to get food at the top of the trees, and the stretching or something somehow gets transmitted to their children, and they develop giraffes develop long necks because of that. And Darwin showed very convincingly that that's not the way it works, that there's selection. You have giraffes with short necks and longer necks, and the ones with the longer necks have more suitable survive. They survive at a higher rate, and so they pass those genes on. But this genetic, this methylation being influenced by the environment and having that effect be inherited across several generations almost sounds like a Lamarckian idea, but it's not. It's not. It only lasts for a couple of generations, and it doesn't involve a change in the DNA sequence. It's just a change in this methylation of the DNA. Okay, so this is an example of what we call an epigenetic change. So epigenetic means it's not a genetic change. It's something that's epigenetic. It's outside of genetics, but it can still affect one's health and susceptibility to disease. Okay, so the point I want to make here is that DNA methylation is laid down during the course of development, and it sometimes is influenced by the environment as well. And then when DNA is methylated, it tends to attract proteins and other factors that tend to shut down the DNA, so you have less gene expression in areas where there's a lot of methylation. And then that also causes histones to be deacetylated, and sort of there's this reinforcing cycle. And if you look what the chromatin looks like, the euchromatin, where gene expression is taking place, you tend to find the nucleosomes, but you find regions that are devoid of nucleosomes, where transcription factors can bind, and then that allows gene expression to take place. Whereas in highly methylated DNA, you tend to find proteins bound that tend to deacetylate the histones and methylate histones, and other proteins are attracted to make the chromatin very compact, and gene expression doesn't take place. Okay. An important example of how heterochromatin, is regulated is X inactivation. Females carry 2 X chromosomes, but one of them is always inactivated, and that takes place by the formation of heterochromatin. So the inactivated X chromosome becomes very compact, and very little gene expression takes place from that copy. And so when you, but which X chromosome is inactivated, whether it's the one that's inherited from your mother or from the father, is totally random. Okay, so one or the other ends up to be inactivated, and you can actually stain cells and see this. There's a histone variant that's associated with inactivated X chromosomes, and if you use an antibody to stain that, you can see this would be a normal individual where there's one active X chromosome you can't see, and then there's this so-called bar body, which is the compacted X chromosome. As I'll explain a little later, there are genetic disorders where there are multiple copies of the X chromosome, and in that case, the extra X chromosomes are always inactivated like this. Okay. Oh, I'll make a point here. This is sometimes there are mutations that are in one X chromosome but not the other. Okay, and if a male receives that X chromosome with the mutation, that often causes a disease. But women who carry one X chromosome have developed or not developed a disease depending upon the way this X inactivation takes place. So this is random, but it turns out that if you look at a woman's makeup, women are actually mosaics. They're in other words, the cells that in one part of the brain or one part of the skin may have a cluster of cells where the X chromosome from the father has been inactivated, where in other regions of the body it may be the X chromosome from the mother that's inactivated. Normally, this doesn't result in any difference in appearance or have an effect. But if it's not necessarily 50-50 balance of the cells. And so in cases where if there's a mutation in the X chromosome that was inherited from the mother, if that is the one that's primarily inactivated in the body, then that woman is not affected by that. There's no disease. But on the other hand, if it's the one from the father that's inactivated and there's a mutation here, and this one is more common, then that woman actually can suffer the genetic disorder as well, although it tends to be less severe than for males. So women are mosaics. The tissues in women are clusters of cells where in one cluster one X chromosome is inactivated, in other clusters it's the other one. And this sometimes shows up in genetic disorders. Okay. Okay. Another important type of methylation that takes place is that within the genome there are certain regions where there are different patterns of methylation in the female gametes, in the eggs, compared to the sperm. And these are genetically programmed and quite reproducible. So, for example, and the regions where this takes place, it's called imprinting. And these are regions where the imprinting is known to take place, where there are genes that are imprinted. And what this means is that, for example, there'll be, in a certain region of the chromosome, there'll be a set of genes that are expressed from the maternal chromosome, and not expressed from the paternal chromosome, because they've been shut off by methylation in the paternal chromosome. And there are other regions where it's just the opposite. There'll be genes that are expressed from the paternal chromosome but are turned off from the maternal chromosome. And one of the examples I'm going to get to, if I have enough time, there's something called the Prader-Willi syndrome, which results from a deletion of the parental chromosome. And it's in a region where the maternal genes are turned off. So deletion of just the chromosome that came from the father is enough to eliminate those genes altogether, and a genetic syndrome occurs. And it turns out that if this deletion occurs in the same part of the chromosome, but it's the maternal one that is deleted, it turns out there are genes that are turned off on the parental, the parental chromosome, and now they're missing from the maternal chromosome because they've been deleted, and that results in a different syndrome. Okay, we'll go over that in more detail later, but you see, it's for genes that are imprinting, where there's a difference in expression between the parental and the maternal chromosome, deletion of the same region of the chromosome can cause two different genetic disorders. Okay, so then the last thing I want to talk about before we get to those genetic disorders is the telomeres. And this is a set of chromosomes, metaphase chromosomes that have been stained, and then the telomeric DNA has been stained with fluorescent probe against those. So you can see all the chromosomes have these sequences that are found in telomeres. The structure that you find at the end of chromosomes looks something like this. There's a highly repetitive sequence that continues for a couple hundred base pairs. And then here, at the very end, there's something called telomeric DNA, where there's a repeat of this TTA-GGG sequence. And this goes on for, there are many repeats. And the reason that, and then what happens is that part of that is single-stranded. And so at the end of the chromosome, you actually have a loop that looks like this, where this single-stranded region invades the double helix here and forms a loop. And that's called a T-loop. So this is at the end of both ends of each chromosome, you have this T-loop. And this acts as a sort of protection, a way of capping the chromosome. Now, what happens is that you had a lecture about DNA replication, I think, right? And I know you did. When DNA replicates, you have one strand, you have continuous synthesis of DNA on that strand, but on the other strand, the lagging strand, you have to have an RNA template laid down, and that becomes the template for DNA polymerase to work. So you have RNA synthesized first, and then DNA is synthesized, and another bit of RNA, and then DNA is synthesized. Does this sound familiar? I'm pretty sure you had this just a couple weeks ago. So in one strand, there's continuous synthesis of DNA. The other strand is discontinuous synthesis. Well, it turns out that the end of chromosomes are a problem, because DNA polymerase requires to have this primer template complex in order to work. So if you have DNA synthesis going up right up to the very end, say to here, and there's nothing to copy anymore, you end up with a double strand of DNA that just stops, right? Well, there's no way to synthesize in this direction, because there's no place for an RNA primer to be put down for synthesis to take place. And so it's a problem. And the only way you can actually synthesize the DNA is you have to have an exonuclease cut this back to make a place where the RNA primer can sit, and then the DNA polymerase can bind there and synthesize that strand. So the problem is that every time you replicate, a cell replicates, you cut back a little bit and synthesize from there. The ends of the DNA get shorter and shorter and shorter with every replication. And so what happens is that the length of the telomere gets shorter and shorter and shorter. And as a matter of fact, let's see, there's an old observation that cells from tissue cells, if you try to take like a skin cell or a heart cell or a brain or an astrocyte and grow that in culture, that you're only able to grow it for about 40 to 60 cell divisions. This is so-called the Hayflick limit that was discovered many, many years ago. And it turns out the reason, what's limiting here, is that many cells don't have a mechanism for synthesizing, for elongating the telomeres. And so with every cell division, the telomere gets shorter and shorter and shorter until it just almost disappears. And that seems to be the limiting factor. It's what prevents the cells from being immortalized. And as a matter of fact, if you take, there's an enzyme that was discovered a few years ago called telomerase that is able to extend these telomeres. And if you clone this enzyme, put it into cells, you can take cells, you can rescue them from cell death, and they will grow just indefinitely. So this enzyme that was discovered has a protein part and an RNA part. And the way it works is the RNA makes a hybrid, contains the complementary sequence to this repeated sequence. And so it forms a template that DNA polymerase can work on. So DNA polymerase can bind here and extend it a little bit further. And then the telomerase moves down and it extends it a little bit further. And it moves in sort of this stepwise process until the telomere is extended. So cells that have this ability can reproduce many times, but many cells don't have this ability. And there's a genetic disease, actually, that I don't think we'll get to it today because we're running out of time, but it's in your set of slides. It's the last slide. There's a genetic disease that that occurs when this telomerase is non-functional. Okay, so this is the last section. I'll go through as many of these cases as I can before. How much time do we have left? Only 5 minutes. Okay. So these are the slides are sort of self-explanatory. So please go over them at home. So Down syndrome is the most famous syndrome that it's caused by excess chromosome 21. This was first shown, the genetic basis was shown in 1959. Here's the karyotype showing extra copy of 21. Turner syndrome is a syndrome where instead of having two X chromosomes, there's only one. And this usually leads to short stature of growth, a webbing of the neck, that can be corrected surgically. Very slow development at puberty, which can be corrected by hormonal injections. But the women are sterile as a result. And this is the karyotype that you see. There's only one chromosome. This is Klinefelter syndrome. This is men. These people with this syndrome are phenotypically men, but they carry 2 X chromosomes. And in these men, there's a tendency to develop breasts after puberty. Their genitals are very small, but otherwise they're phenotypically men. Both of these diseases, or many of these diseases, are not fully penetrant in all individuals, which means that not all people with Klinefelter syndrome have this extreme kind of symptoms. But in the extreme cases, this is what occurs. These are some just additional ones. The trisomy 13 and 18 are lethal, so babies die even if they're born. Jacob's syndrome received a lot of press a few years ago because the first surveys seemed to indicate that there was an excess of people in prisons. These are phenotypically men, but they have 2 Y chromosomes, and the first surveys that were made seemed to find more of these people in prisons. So there was an association of this with criminality. But it turns out that's not the case. Subsequent studies have shown that this is not, there's no correlation. And here's an example of triple X syndrome, and there are even cases where there are 4 or 5 X chromosomes. This is Cri-du-Chat syndrome, and that results from deletion of the short arm of chromosome 5. And this leads to a variety of problems, but includes severe mental retardation and sort of behavioral problems. This is Smith-Magenis syndrome. This also, we're actually working on a little bit on this in our lab because it contains a gene that's been linked with autism. And so Dr. Chen Li, who gave you a couple of lectures on the last course, is actually working on this retinoic acid-inducible gene. This is a dose-sensitive gene, so if you have too much or too little, it causes problems. This is Charcot-Marie Tooth syndrome. This is one that causes, this is called stork feet phenotype, or stork leg, where there's a wasting of muscles in the lower part of the leg. The upper part is not as severely affected. So you have these stork-like feet, or legs. You have this arched feet and clawed toes. And this is a neuropathy. So there's a problem with the peripheral nerve. And this is actually caused by a duplication of another dose-sensitive gene, which encodes a protein that is in the myelin sheath of the nerve. So if you have too little of that, this is what happens. This is another syndrome that we're interested in. Deletion in this region causes a lot of problems, but we're interested in it because 20 to 30% of these individuals develop schizophrenia as adults. And so one of my graduate students is looking at one of the genes that's in this region that we suspect is a dose-sensitive gene. This is a very strange syndrome where you have unequal crossing over between the X and the Y. And there's this SRY gene is the gene that determines maleness. So if you have this gene and it's active, you're a male. If you don't, you're a female by default. And so sometimes you get unequal crossing over where this SRY gene is transferred to the X chromosome. So the person, two X's usually means you're a woman, but if you've acquired this SRY gene, phenotypically you're a man, and but usually infertile. The Philadelphia chromosome is a non-equal translocation that causes a fusion between a kinase called ABL and then some other gene. And the effect of this is to make the kinase constitutively active. And when that happens, it causes the cell to grow. And that's why the cells become cancerous. So this is a translocation that's seen that occurs independently at a pretty high frequency, and it's a common cause of this chronic myeloid leukemia. The Prader-Willi, do you have to go right away, or 2 or 3 minutes? 2 or 3 minutes, okay. The Prader-Willi Engelmann syndrome can arise from unequal crossover, this involves one of those repeated sequences I talked about. When people who inherit this deletion that's missing this region, here the genes, these genes are expressed in males, and these are silenced in males. Here it's just the opposite. These genes are normally silenced, and these genes are expressed. So the idea is that if you have a deletion of these genes, and the deletion takes place in the parent, from the father, you get normal expression here, but you don't get normal expression there, and that is the cause of this Willie-Potter syndrome. One of the phenotypes of this is uncontrollable hunger. So these people cannot stop eating, and they become often grossly obese. So sometimes, you see pictures of very grossly obese individuals. Many of them have this syndrome. Okay, and then but deletion in the same region, but on the maternal chromosome, causes Angelman syndrome, and this leads to very stereotypic behavior. These kids are going around like this, and they're very happy and smiling. My first graduate student's PhD student's son has this disorder, by the way, and so it's not that uncommon. But you have to see, it depends upon whether the maternal chromosome's deleted or the paternal. You get Prader-Willi. If it's paternal chromosome that's deleted, you get this syndrome if the maternal one is deleted. And then this is the last slide. This shows the structure of the telomere. This is the set of proteins that are important for extending that. Mutations in any one of these gives rise to this dyskeratosis congenita disorder. And what happens is that you get tissues that are growing all the time, like your nails, for example, run out of telomeres and they start to die. And so one of the early symptoms are problems with the nails. Another early symptom, there's problems with the skin, the white patches on the tongue. But the most important one is that the blood cells that are constantly normally being replenished can't be replenished because the telomeres have disappeared and they die, and then the patients die from anemia. Okay, so that's it. The rest of the slides just has to do with reference materials. Okay, so that's the end of the lecture.